Okay, now we are going to discuss the meristem culture for virus elimination. First of all, this process was developed by Morel and Martin in 1952 for in vivo virus eradication. So, shoot of all angiosperms and gymnosperms that are grow by virtue of their apical meristems. The apical meristem is usually a dome of tissue that is located at the extreme tip of the shoot and measure approximately 0.1 millimeter in diameter and 0.25 to 0.3 millimeter in length. Meristem, or which is also known as the shoot tip, is isolated from the stem by applying a V-shaped cut. Then MS medium salts have been very satisfactory for such cultures through whites and were the most widely used mediums during the early days of meristem culture. So here is the apical meristem. This is the portion which is used in meristem culture. This is the flow diagram which is indicating the morphological view of the shoot tip. Then excised meristem is grown in the culture medium, and then here is the rooting, and then this is supplemented with the soil, and here is the plant led formation. So where there are various stages for the meristem culture. In the stage 1, culture is establishment stage, and when X plants may develop into single shoot or multiple shoot. At this stage, X plants are supplemented with cytokinins like BA, kinins, and 2IP. In stage 2, the objective is then supplemented to multiply the propagules, and for this axillary shoot proliferation is followed as it maintains higher genetic stability. In axillary shoot for proliferation, high levels of cytokinins are utilized to overcome the apical dominance. In stage 3, purpose is regeneration. This stage purpose is to regenerate the adventitious roots from the shoots which are obtained in stage 2. Numerous studies have indicated that NAA is followed by IBA, IAA, and 2,4-D and other auxins which are used for the induction of root generation. Different components of mediums are used, which includes the inorganic nutrients, that includes the nitrogen, phosphorus, calcium, magnesium, and sulfur. There are also carbon sources, organic supplements, which includes vitamins, amino acids, different types of complex organic compounds, and other plant growth hormones such as auxins, cytokinins, gibberellins, abscisic acid. Then there is also a solidifying agent, which is in most of the cases agarose. pH, which is very mandatory component of the medium, that should be around 5.8. Lower than 4.5 or higher than 7.5, that greatly inhibits the growth. So the ideal pH which is considered good for the tissue culturing is 5.8. Protocol, first of all you have to remove the young twigs from the healthy plant, then cut the tip 1 centimeter from a portion of the twig. Then surface sterilization is very important step in the tissue culturing. Then surface sterilized X plants are transferred to the petri dishes. Then outer leaves of each shoot are removed, and after the removal of leaves, the apex is exposed and cut off the ultimate apex with the help of sterilized scalpel, and then they transfer these into a less than 1 millimeter in length. Then incubate this culture for under 16 hours light at 25 degrees centigrade. As soon as the growing single leafy shoot or multiple shoots obtained from single shoot tip or meristem, transfer them to the hormone free medium to develop the roots. Then plants are later transferred to the pots that contains the composites and kept under greenhouse conditions for hardening or for acclimatization. There are various applications of shoot tip or meristem culture. First of all is in the virus elimination. Plants are often infected with more than one type of viruses, including some even not known viruses. So a general term virus free is used by commercial horticultural by this method. Second is micropropagation, asexual or vegetative propagation of whole plant using the tissue culture techniques referred to as micropropagation, and this is achieved through the meristem culture. If you want to store the genetic resources, then is also very important process. This process is also used in plant breeding to form the pure lines or pure hybrids, pure non-viable. If in case of if there is any hybrid plant which produce abortive seeds or non-viable seeds, as a result it makes barrier to crossability in plants where non-viable seeds are unable to develop into mature plants. So shoot tip or meristem is used to culture such tissues or such plants in the breeding programs. Quarantine plantlets derived from shoot tip or meristem cultures are easily accepted by the quarantine authority for international exchange without any checking. So using this technique, crop plants can be easily exchanged in crop improvement programs that are based on materials from different parts of the world. Meristem culture is exploited for various purposes. First of all, used in eradication of banana virus from infected banana plants and production of certified banana virus-free banana plants. It is also used for the elimination of viruses through chemotherapy and meristem tape culture and nectarine. Production of virus-free chrysanthemum is very important. Then pathogen free other than virus like fungi, bacteria can also be got rid of using this process. Baker and Phillips in 1962 obtained carnation of plants free from the Fusarium roseum species. Similarly, another scientist obtained plants free from the Oxysporium species. Advantages of this process is that this process lack the vascular tissues. There is higher oxygen concentration. This process is also used for the production of virus-free plants. This also help in the facilitation of exchange between locations. Cryopreservation is also another advantage of this type of tissue culturing. First, a disadvantage is that the isolation is very difficult. Meristem tissue shoot tip isolation is very difficult. The survival rate is very low, and regeneration time for different explants may vary or may be long. Maybe it takes around 8 months in case of potato plants. Removal of explants causes a setback in the growth of mother plant. Means if you are removing any meristematic tissues, then there is some drawbacks with the growth of mother plant. Thank you.